Okay, my friends, shocker du jour alert. Stay tuned. 240 million year old dragon found in Triassic rock stuns the world. Well, let's take a look at it. Okay, you know I'm a little obsessive about details, and that's what I require. I require details. I have a lot of people sending me things, drawing pictures over. You can see this, everybody can see it. Well, if you can't see the details like this, then you don't have the details. What is in this details, though? You can see it's got a big long neck, it's got a head. Everybody, I think, looking at this, if they accepted this as being truly real, they would have to say, yes, this is more than likely some kind of a thing with a big long neck. And here's I look for the details like this. What is this? What's all that stuff? Oh, those are ribs. No, they're not. The ribs are here. All right, let's get in and look at it close. This is what I do. I look at the details, and if I can find the details, then I know what I'm doing. These are ribs. This is not ribs. That's something, I don't know what it is, but I would suspect it's some kind of feathers. They had feathers. They had scales. They had everything. And guess what? I have feathers here from something much bigger than this. And I also have a dragon scale here, which would be from a very, very large dragon. And the feathers from a large feathered creature. And this, my friends, right here is a feather. You see that? That's the thickness of the feather. And I think it says it's like a turkey feather. It's damn near the same color. You see that? That's on one side. And here's the other side. All right, and I'll show you where this came from. My friend, the stuff I showed, and I will be showing this artery. That thing right there is an artery. That's an artery. Wait till you see. I show you the details. No question it's an artery. And there's no question this is a feather. I'll show you the huge feather it came from. This is the thickness of that feather. This is the thickness of a normal feather. There's almost nothing there. And let me show you that. And we're going to be talking about this dinosaur, dragon, whatever you want to call it. But it's in Triassic rock. Now, I am in the Triassic area, and I have the same stuff. But mine is completely fleshed out. Not like this. Mine is full-body creatures. Like this goose, my buddy here, Caesar Augustus. There's his little nose hole. There's his eye. He's a very serious guy. There's his feathers, and he died laying in a flood like this. And his neck pattern is in here. If you look at this and you know how to shadow things and pay attention carefully, you can see his neck. It's right there. Plus his artery is here. The vein is over on this side. And he died in the flood, just like this lung. Hold on. This lung, I had DNA tested and CAT scanned and everything else. It's a human lung, and he died in the flood too. Flat as a pancake on the back. And I have so much of this stuff, I could fill a museum easily. And it's all from the Great Flood. And this did not happen that long ago. I am in an area that's called the Central Connecticut Rift Basin, which means it's an inland bowl, and it filled with water boiling during this event when we almost got impacted by a fiery comet, which boiled all the waters in certain areas and pretty much all the whole world, and created the Triassic Great Dying. Everything died. There's no dispute about this. They agree with that. It's called the Great Dying, and it's the end of the Triassic era, and everything on Earth pretty much died. And then after that came the dinosaurs. Well, everything I have here, like a goose, that's not a slimy little creature. They say there was nothing with bones or teeth or shells. They were just slimy little creatures that all died off. That's just not true. There was all the same creatures we have here today, basically. All right, so And I have all the stuff to prove, and I have had my stuff DNA tested and CAT scanned and everything else. And the problem is it won't be examined. And now there is no excuse, as my new song says from Harold Peterson. You have no excuse. Now, let's see where this came from, this feather. Now, this thing here I think it's only 16 meters long or something, which is what, 50 feet, something like that. That's not what you consider much of a... Actually, that would be a parasite. On the creatures I'm serious, and they wouldn't even know it was there. All right, before we get in a little deeper, let me just show you what he has to say about it. This is a channel called Trending Mailbox. 240 million year old mysterious Chinese dragon discovered. All right, now, it's only a minute and a little bit longer. Here goes.
Team of paleontologists have revealed an extraordinarily well-preserved fossil of a strange aquatic reptile that lived approximately 240 million years ago during the Triassic period. Nicknamed the Dragon of Lingwu, after the region in China it was found, the creature has been identified as a new species called Dinocephalosaurus orientalis. The fossil measures over 16 feet long, with a neck spanning over 5 feet.
It's only 16 feet long. That's bacteria size compared to the things I'm talking about. Anyway, let me just finish up.
Longer than its body and tail combined. Dr. Nick Fraser, curator of the National Museum Scotland, who helped study the fossil, told BBC that this creature had flipper-like limbs and an extremely long neck with 32 separate vertebrae, making it a very strange animal indeed. This discovery allows us to see this remarkable long-necked animal in full for the very first time. It is yet one more example of the weird and wonderful world of the Triassic that continues to baffle paleontologists.
Boy, you're not kidding. Now, I'm going to just back up here a little bit and get out of this. You can come up and watch this if you want. But this is, I believe, these are the feather stalks, the center of the feathers. These things would go away, but you'd have all these little stalks hanging around. I believe that's what I'm taking from this. And what would they be? What are these six things doing here? They were there for something. All right, those aren't from the ribs. They're not from the ribs. Not in my mind, they're not. You see them? They're all over here. And then there's none on the tail. So it was like a feathered thing with a snaky tail. I mean, there were all kinds of different dragons here. I've shown Quetzalcoatl, I've shown Typhon, Scorzilla. You know, they were not uncommon. They were the norm, literally. And this is the Triassic when the Great Flood happened. 3 layers: red bed, gray clay, black cap. And that's because they boiled all the flesh off and made the red bed in a muddy slurry. And then the gray clay was all the organs and the tendons and the muscular stuff that dissolved next and came down onto the red bed. And then the black fallout was from the volcanoes and all of the burning and all that stuff settled. So you had red bed, which is the flesh, gray clay, which is the rest of the body parts, and then black cap on top. And where I am, it appears that there was a very interesting way that it happened. It must have killed the things fast and preserved them quick, and a lot of silicates, and it just sat here in a bowl and cooked them until they just all turned to stone. Now, for some reason, Caesar, everything's here, his feathers and everything. But most of the stuff I have, like the lung here, everything that this guy had when he died, and I could tell you how he died. He died and his whole body was there laying on the ground. And how can you say that, Roger? Well, here's how. Because this is as flat as a pancake. And you say, so what? That doesn't mean his whole body was there. I say, well, how about this? That's where his heart was. I say, his heart was still here. And it was going thump, thump, blah, blah, dead. And then it just sat there cooking. And all of the flesh melted off to become the red bed and the clay. That's clay. That's all it is red clays are red flesh that's eroded. And then all the bones dissolved. There's no ribs or anything around it. The bones dissolved. Plant life dissolved. Trees dissolved because of cellulose. Creatures cooked. They either cooked and boiled off into soup, or they sorted their parts out into organs here and there, because the lungs and the hearts and the livers preserved fabulously. Lungs, I got it coming out of my ears. There's another lung from, I don't know what it's from. And then the silicates fill in the holes. If there's holes, the silicates fill them in. If there's no holes, they just bond with the residents that are there, and they turn it into pretty much aluminum feldspar, aluminum silicates, which is basically on all rocks. Like, that's all aluminum silicates too, because all the coatings and the membranes are made out of phospholipids. Phospholipids boil down into aluminum silicates. This is where you need to know a little bit of chemistry. Phospholipids, phosphorus lipids, fats, boiled down in this soup down to aluminum silicates, ALSI. Every feldspar is an aluminum silica, and every feldspar is a membrane. All right, I hope that makes sense to you. And the bones and everything just turned into stone because of a process called nucleophilic invasion and substitution. I wrote a paper on this in 2015, and it's still pretty valid right now. It says exactly what was happening to these creatures to petrify them. Now, this is strange because it seems like everything was stripped away, or they chiseled every little bit away, because you could chisel everything away and end up with what was inside there, which would be the bone. Because originally, if you had this, it would be covered with flesh, and my stuff is the whole. Well, here's one. Hold on. I'll give you two examples right here. One and two coming up. This, my friends, is a no-toe foot. All right, there is bones and things in here. There's different stuff in here. I don't know if it's bones or what's in here, but there is the same sort of stuff we have in our feet, basically. And if you chiseled this away, or if this was in some kind of condition where this didn't petrify as perfectly as it did. My area here, everything I have petrifies absolutely flawlessly, except on occasion you get something like this, which is something like they're showing there. It's everything's boiled out of it, basically. And this is an apical tuft from a finger. And I'll show you another finger I have from the same hand that didn't boil away. Anyway, if you chisel this away, you'd get down to where there's bones in here. And maybe that's what they did here. They found a bone and they said, ooh, and they started chiseling away, and they said, ooh, there's another one, ooh, and they just kept going. You know, as long as there's funding, they'll keep chiseling away. Now, this is a fingertip. That's a fingertip. And that's been CAT scanned and DNA tested and everything. It's a giant human fingertip. And I actually did what they might have done. I removed this piece here. It got down to the bone. This is the bone. All right, there was a piece right here, and I still have it. But I got down to where the bone is, and you can actually see the bone is there. And just like these things are here, but you got to chisel all this stuff away, and I could do that. I could chisel until I got down to what the bone was, because it's there's going to be a membrane between it, and these things come off according to those membranes. So they may have chiseled down into this, but I would have suspected this to be more fleshed out than it is. But there is different chemistry happens. Sometimes things change in all kinds of different ways. Like even this one here, look, that's a piece of meat, and not literally a piece of meat. And it's got the fascia, and it's got the tendon in it. It's got all the marbling. So it depends on what condition it was in and how long it was in the waters for, how high it up, how quick it dried out. Because the ones that dry out quickly, they fall apart. The ones that were deep in the lower end of the water pool, they seem to have held up very well. And my area here is just, they're just flawless. I mean, they're just flawless. It's absolutely incredible how detailed and perfect. I mean, the bones and everything. The cartilage, that's the cartilage right there. And that, I believe, is where I think those are the nerves run up. Maybe there's a vein and artery, I'm not sure. But that's the marrow area inside, and there is still a taste of bone left here. I'm going to show you that, because that's bone. The rest is turned to stone. They say, oh, no, if it's stone, it was never a bone. I said, no, incorrect. It was a bone. It was nucleophilically invaded by whatever the stuff was around it. And the molecules filter through there, and eventually they find stability with whatever is predominant in the area. The thing that is dominant will take over and just change it to whatever it is. Sometimes they turn it to opals. That could be an opal. This could be like this. And most of them are. Most of them are. They don't, they don't. Anyway, I think you get the point. This is an anomaly, and I think they chiseled away at this stuff to get down to where they had this perfect fossil on. This would be the gray clay. Now let me show you one more thing, and then I'm going to get into my stuff. All right, I'm pretty sure I showed you the feather. And this is a huge pile of feathers on the side of the road. People drive by it every day, have no idea. He smashed this off, or broke it off. But I believe this was a feather laying up against another feather, and that's what this came out of right there. I'm not sure exactly the orientation it came out of, but this is, you see it? You see the exact same? It came right off of there, so it's obviously it's going to be the same, and it's the same colors and everything else as a turkey feather, but it's a lot thicker than your normal turkey feather. And both of that, this and another thing I'm going to be showing you, or have shown you, which is the artery, both came from Greg. Greg is a fabulous source, and I have a couple other things to show you of his. It won't be in this video, but and I have things people send me from all over the world, and and a lot of people ask, can I send you something? And at this point, no, please. A picture would be just fine, but that's about it. I'm just overwhelmed. So anyway, and I have everything that's virtually indisputable. Gigantic things, huge things. But it's too much for the human mind. That's the issue. It's not that I don't have the evidence. I have proof. I have DNA tests. I have CAT scans. I have the specimens. I have the blood. And it refused to be examined because it's an academic disaster. That's the issue. All right, if you've been around here, you've seen this dozens of times. This was no cheap, quickie swab test. This was drilled stuff inside, meticulous harvesting, and sent off to be tested. 3 different samples, 3 of them. And they did the extractions. They took bacterial DNA, which we're loaded with bacteria. So they have to take all of that DNA out of your body and then clean it by extracting with certain primers. And that extracts the human stuff. And it tells you what's in there. Now, I took it from deep inside there. So all of the bacteria stuff they get out, and then they see the human stuff, and then they match it to what is in the national or in a worldwide database. And this is how it was all done, how the extraction was done, how they bonded the things and did all whatever they do. I don't really, I got to be honest with you, I'm sort of speaking over my head because I don't fully understand this. This is very technical, and it took 3 months to do, and it wasn't cheap. It wasn't, I could afford to do it, let me put it that way, because I wanted to prove this, and I wanted to prove it. And it came back, these are the different things, and these are the different primers they used, and there was a couple different sets of primers, and one of them had excellent quality DNA. With those primers, they showed up excellent quality DNA attained for the 36-inch tip and the lung. The lung is the thing I showed you here, right there. All right, and the 36-inch tip is a 36-inch long finger tip. Now, I drilled these things out and sent it to him. He didn't extract the DNA. I extracted from blood, sent it to him. They submitted it to Union, New Jersey, to the Eaton Bio Labs, and they sequenced and did all kinds of things, whatever they do. And then they put it through the National Center for Biotechnology Information Database. Each DNA sequence was matched to all DNA sequences contained in the sequence database. And this is what it came back as for the results of the blast. Homo sapien mitochondrial cytochrome B gene and D loop region, both of them. These two were the most densely, the mud fossil, the mud tip was not quite as dense with the DNA, so they just bothered to show these. But then these are all CTAGs, the DNA sequences, to prove that this was mitochondrial DNA, which is human mother side DNA from Homo sapiens. And I said, is that a modern human or a Homo erectus or something? He said, that's a modern human. And he said the last time I talked to him, he has had some people going against him for doing this. You can't even find anybody who will do this, because they attack you for even doing it. And I did talk to him finally, and he just said, be certain that everyone knows that you were the one that extracted the sample. So if I sent in some fake stuff, well, I faked it. I did not fake it. All right, he says, I'll stand behind the results. The results are real and absolute for certain. He said, but make sure that everybody knows that Roger Spur, not this guy here from Helix Biolab, was the one that actually took the samples out of here. All right, so I drilled the holes in these here inside to get the blood out. I clean them up real good first. You drill out, you get it clean, and you drill deeper, and you take that stuff and you send it off. Put it in glassine bags to double bag it, and off it goes. Of course, you mask up and do all that stuff, gloves, but it doesn't matter. It was dense. He said it was excellent quality. He said it was dense. He said it was just like blood, and it was. I know where to take it. I took it right out of the artery. And where did I show you this artery that Greg sent me? The thing is as big around as my leg. All right, here it comes. Okay, this is a much more complete fossil found. They claim it's 240 million years old, found in Triassic rock. And here's what they have to say about it.
Allowed paleontologists to fully analyze its anatomy for the first time. As Dr. Fraser noted, every time we look in these Triassic period deposits, we find something new. This finding provides more insight into the odd menagerie of marine creatures that filled Earth's oceans after the Permian mass extinction event 250 million years ago. The lead researcher speculated its lengthy neck may have given Dinocephalosaurus a hunting advantage over other aquatic reptiles it competed with.
All right, so they're assuming that this is, actually they're talking about being a Chinese dragon. And it's 16 feet long, I believe, is what they said was the dimensions. All right, this is 240 million year old mysterious Chinese dragon discovered.
Team of paleontologists have revealed an extraordinarily well-preserved fossil of a strange aquatic reptile that lived approximately 240 million years ago during the Triassic period. Nicknamed the Dragon of Lingwu, after the region in China it was found, the creature has been identified as a new species called Dinocephalosaurus orientalis. The fossil measures over 16 feet long, with a neck spanning over 5 feet, longer than its body and tail combined. Dr. Nick Fraser, curator of the National Museum Scotland, who helped study the fossil, told BBC that this creature had flipper-like limbs and an extremely long neck with 32 separate vertebrae, making it a very strange animal indeed. This discovery allows us to see this remarkable long-necked animal in full for the very first time. It is yet one more example of the weird and wonderful world of the Triassic that continues to baffle paleontologists.
Yes, it does, because I think I can shed some serious light on this. And the Triassic was 3 layers. There was a red bed, gray clay, and a black cap. I would say this is on the gray clay. You see how flat that is? And the reason this is flat like that is because this was the great dying, the great flood. The red bed consisted of the flesh of the creatures, the red bloody flesh. The gray clay was the rest of the body parts, the tendons, the bones, the this, the that, the organs. They dissolved as well. And then they covered the red bed with gray clay. And then there was a black cap that came down, because this was almost an impact by Venus onto the Earth. Created huge destruction on the Earth, absolutely just devastating. Velikovsky recorded all this and he has it in a book called Worlds in Collision. Look it up. Worlds in Collision. Immanuel Velikovsky. He was the hero that researched all of this and knew about this great dying, but it didn't happen 250 million years ago. I am right in the Triassic era, and I have Triassic things right here that I will show you. They're Triassic, from the red bed, the gray clay, and the black cap, and that all happened in almost instantly, the same things. And I will show you I can prove that, because my gray clay is pushed down through the red bed with a human footprint in it. I'll show you right now. Okay, this is Nero's bone. This is my dog. And I cut it across here. Nice and flat, so we can see what it's like. And it is absolutely nothing like the artery that I just showed you. Now I'm going to come right down on top of it and see what it looks like. It is just absolutely 100% different than the other bone, the other artery that I showed you. It has none of the same characteristics whatsoever. And there's the cavity way down inside here. Oh, it's too deep to see. Well, that's inside a marrow. You have all this webbing. As a matter of fact, hold on a second. All right, you see Nero's bone here. That's what's going on inside a bone. This is a bone, marrow aspiration, they call it. They're going in to find out why you're not getting enough red blood cells. This is where they're all manufactured, inside your bone marrow. Well, then they're transported through your body through that gigantic artery. All right, that is the surface of the artery. All right, that's the surface. And all of those little bumpy looking things is what locks it in position in your body. All those things grow into that. And that it's not going to move around a whole lot. They stay where they're supposed to stay. Now that's where it's wet. Here's where it's dry. Oh, come on over here. All right, this is what it looks like when it's dry. All of those little bumpy things are what holds it in position. And they all turn, these are silicates. There's a lot of silicate here. And that's because everything was affected by the siliceous ooze that came off the ocean floor. And it bonded with all these other metals and minerals that were in the body and fused. Mostly it's silicon dioxide, SiO2. But then you have phospholipids, which were the membranes. They turned down into aluminum silicates, which are feldspar. So the silicates play a huge, huge role in this. Okay, as far as I can tell, this right here is just completely red blood. Now I'm going to just squish around a little bit here and see if we can turn it back into blood again, which we certainly can. You see this? This is just now turning right back into blood. It's just turning right back into mushy blood. I can just keep going deeper and deeper and deeper with this, and you get more and more and more bloody, because this here on the outside is already fused. Look at this. Look. There it is. There's the mother lode, baby. You want to see something happen? We're going to put some catalase on a hydrogen peroxide on there. This baby should explode. Where'd that go? Where'd you go? It's all over the place. No, it's real. I mean, I could just dig way deep in here if I want, because we're right down into the actual blood cells. Now, I guess I'll put a little hydrogen peroxide on there. Let me give it a minute or two to dry up somewhat. Because here's the deal. Hydrogen peroxide is H2O2. Water is H2O. So there's only one O, oxygen, in water. Well, that evaporates pretty quick. So you end up with the H2O, water evaporates. All right, so we want to get that water out of there. So then we're going to put some H2O2 on there instead of water, H2O. And inside of anything that's living, there's what they call catalase. Catalase is an enzyme, and it will attack the hydrogen peroxide and take that extra oxygen and say, you're not coming in here, get out of here. And it'll bubble up like crazy. Now, I'm going to let that dry up for a minute or two. It really doesn't have to. I could put it on there now. We should see a very, very strong reaction. I'll go off the wall if this doesn't give us a reaction that's going to be just intense. I mean, as far as I'm concerned, that is raw red blood. And the minerals have turned into silicon, yes. These little silicon bits, true. But the rest of the red blood is in there, the plasma and so forth. We should get a very strong reaction. That's what I'm hoping for. Okay, that looks pretty well dried up for, not dry, but it's dry enough to get a good reaction, I believe. Again, this is all red blood in here, and red blood has the catalase, and the catalase reacts with this hydrogen peroxide. Wow, it's just, watch it get real foamy. See it bubbling around up here? See it all bubbling? This is just nothing but bubbles, but I have to get right down on it to see it, because there's so many different particles here. It's just one big mass of foam, watch. You see, it's whitish looking. These are actually bubbles. That foamy looking white stuff is bubbles, but they're so fine because every single blood cell is giving off this catalase reaction. Let me see if I can get really right down on it. That's about as far as I'm going to be able to get. You see them bubbling? It should take, it looks like it's taking longer than I thought. But I'm seeing, I know what to look for, because these are just like you'd see in membranes. It's a very, very fine foam. And you almost can't tell it's foaming. All right, let's see if we can get a reaction down inside here. Hold on. All right, I'm going to put some catalase right down in the middle here and see if we see anything happen. I'm not seeing what I thought I'd see. That's unusual. I would have expected to see a strong reaction. Which we are not seeing. When I say a strong reaction, it should be bubbling. I'm going to really hose it down here and see what does happen. Maybe it's just... All right, now we're seeing it. There's just so much catalase that it was wiping out the extra oxygen is too fast. Keep your eye on it, just because it's got to soak in for a minute. This is just, there's so much catalase that the reaction is too fast. But I'm going to tell you what, I'm going to go out to one of the membranes. We should really see it react in the membrane. Membranes always react very, very well, because that's really where the source of the enzyme is. Now that right there, that layer right there should give us a real good response. All right, let's find out. I got to be honest with you, I'm shocked that the blood didn't give us a better response. But this is the kind of area that is going to be where this enzyme is, because this won't let the extra oxygen come in to hurt you. Let's see if we see it foaming up. We're sinking right in. Very little foaming. Boy, this is strange. Very strange. Seen a little foaming here and there, but not what I would expect. That might be foaming of a membrane. You see it's changing there? Huh. Quite strange indeed. I wonder about the outside layer. Boy, this has got me going here. Something's boiling away over here. There's a little artery or something going on over here that's bubbling up. But boy, I'll tell you, I should have seen all kinds of bubbling. Very, very strange. Something is going on there. Whatever that is, I do not know. Let's put a little more in there. These Q-tips that I'm using are coming out like you just took a clot of blood. Let's see. Up over here, let's come down and look at that. See, it's down in here. There's some catalase down in there, but we should be seeing it everywhere. I am just shocked. Although this foamy stuff is also catalase, but it's such a fine amount. This is some kind of little artery or something that's down in there, or some kind of little tubule. I'm going to look at the outside of it, because this has got me very, very interested. Quite interesting. That one here keeps coughing up something. right, now hopefully you can see this. There's a little divot in here. It looks like it happened when this thing was being petrified. You see it right there? That little gouge. There's a crater, like crevice going across. We're going to look at that and the transition between the two and see what we find. Now, hold on. We're going to go. So don't forget, that's what we're looking at. And now we're going to look at it up here in the microscope, because there is a gouge there. And we can see that gouge. All right, let me turn the lights off so that we can get a better shot of it now. All right, here's what you're looking at. This is down inside the gouge. All right, so we're way down inside the gouge. Now, if we come back up or go this way, we can go here to see the top layer. All right. And this is scraped away, so now we come back and go down inside, and we're down here. So let's put a little catalase on this and see what happens. So we've got two different areas right in the one spot. Let's see what happens, because I'm kind of surprised it didn't happen on the other part. And I'm wondering what could have wiped all that catalase out. I've never seen that happen before in something that has so much blood and so not amount of catalase. It's got me quite interested. Now this is quite a ton of catalase. See it? Watch, I'm going to come. This is what I had expected to see down in where the blood was. But this is actually more going to be more active now that I'm thinking about it, because this is the membrane. They're supposed to, primarily they're in membranes, so they don't let the oxygen in. Now I'm starting to think, I'm starting to use my brain. This is where you would expect it to be. I was thinking inside the blood. It's more before it gets to the blood. Let's come to a clean spot right here. I'm going to put a batch of it in there and we'll take a look. And don't forget, the outside of this artery is going to be the membrane, because that's where it goes to separate that from the rest of your body. So here's where you see it boiling in there. You can actually see it boiling away. That's why I was saying that you got to get right on top of it to see these tiny, tiny bubbles. You may even still have a hard time seeing them. They're coming out profusely. But they're so, it's like a film of like a white haze. But you can actually see it boiling away if you look really careful here and there. And it's eating up all the extra oxygen. So it's going to diminish its reaction, because it's eating up all the oxygen. Once the oxygen's gone, it won't bubble anymore. So you got to almost continuously put more and more. Well, you do have to continuously put more and more if you want to get a good reaction. All right, see it now? See it bubbling? The more I put on there, the more it will penetrate into that membrane and get fuzzier and fuzzier. All right, just one or two little hits is not enough, because it's so strong, this catalase. Let's see if I can back this up. See, we're actually down in the membrane there. This isn't boiling anymore over here, but down here, that's why it's so fuzzy looking. It's not because it's out of focus, it's just fuzzy looking because it's boiling off the little bits of bubbles. You really got to look close. Let me see if I can get any finer. This is where it gets hard when these little, when you really got to get close. That's about as close as I'm going to be able to get. I'm going to do one more pop because I'm pretty well focused in now. Hopefully I can get it right on the spot. Now what I want you to do is watch for over in this area here is a good area that has a lot of catalase, I think. Okay, let's watch over here. You see that fuzzy looking white? That's from the catalase reaction. If you look, you can see it boiling around here and there. I have seen much stronger reactions, I have to be honest with you. This thing here, I did notice there was very highly silica inside of it, so it has transitioned quite a bit. But I mean, you can see it's an artery, gigantic artery. Doesn't have the reaction I had expected. I got to be perfectly honest with you. I can see it, but it's not what I would have expected. It's not strong enough. You know what? Let me try one last thing. Let me scrape this up a little bit. Let's put another taste of hydrogen peroxide on there. Yeah, you see, it's better, but it's not fabulous. Certainly nothing to write home about. There's literally raw blood coming out of there when I dabbed it. Like this stuff here, look at this. That's dripped off of there. It's just like raw blood. Now, I guarantee you they get DNA out of that. There's no way in the world they couldn't. But I got to be honest with you, that catalase non-reaction was not expected. So that's what research is. Getting what you expected and sometimes getting what you didn't expect. But what I did expect is this is not a bone. This is a bone. That, my friends, is an artery. And that I smoothed off so that I could look at it. I don't think I showed you that. These have all, well, let me show you. All right, here we are looking at this bone. I have a spot that I ground smooth so we can see. The rest is just a natural, preservation in the ground, apparently. Now, I'm going to show you how fibrous this stuff is. So we're looking straight down into the vein, into the artery, the construction of its surrounding tissues. And here it is here. I have to focus it in. So let me first, you know what? I'll bet you I get catalase reaction out of that. I'll bet you I do. Right there. If I don't get a catalase reaction out of that, I will eat this rock. No, actually, probably not. But I will, although you probably could if you could somehow grind it up enough. I mean, it's got all the same nutrition. Now, let's see if we can get a catalase reaction. Hold on. Let's turn this off. Let's focus this in. Let's get lights a little lower here. All right, now this is the place I would look for, up here and over there. Let's see. Well, we'll just have to see what we get. That's the fun part of research. You just never know. You expect and you suspect, and then you see what you see, which is nothing. Boy, I'll tell you, this is odd. This must have been in something that killed the catalase. That's just not right. Sacre bleu. Come on, something. Come on out of there. What's the matter with you people? There's a little bubble here and there, but that's just not right. That should be foaming. This had to be in some kind of chemistry that wiped out the catalase, which means it wiped out the bacteria, because the bacteria create the catalase. Interesting. Well, maybe that's why it's so dried out. And it just eventually died, because most of the stuff I have has been preserved in the mud and then taken out of the mud, and the catalase is still very reactive. Very reactive. All right, let's take this from a different approach. That thing there looks like it's been out of the ground for a long, long time. To me. And it's really sandy and gritty, and it doesn't have like a mud fossil effect to it. Mud fossils are almost soft and smooth. That's like sandpaper. That is tough. Now, and I can't find any catalase to speak of. So it was in some kind of a condition that caused it to end up like this and destroy the catalase. Now, this is just a regular body part. I'm not sure what it is. I believe it's some kind of an organ. But it will have a membrane. You see how you got this brown layer and then you get into the black? Let's see if we can get a catalase reaction out of this. Because we should be able to along that membrane especially. All right, this time we're going to be looking at that rock right there. It has a membrane around it. And it's two different shades. You can see the difference now. I'm going to go up into the other screen, which is we're going to be looking right at that spot. So here we go. Now I got to turn the lights off. All right, and I got to turn this one off. All right, now, so whatever this organ was, I don't know, but it has a membrane. And inside is some kind of meat, whatever it was. A liver, or who knows what. But this is where we should get a pretty good reaction. So let's see. See it? Look at that. Look at it. That's what I was expecting to see from the other one. I'm telling you, I'm still freaked out. But this is what you should see in a membrane. You see that? Look at how reactive that sucker is. Look at that stuff. You don't see that happening over here because that's where the catalase is, over in here where Look at that. I don't know what happened to that other one. It's going to freak me out. I'm going to be working on that for the next week. There had to be some chemistry that wipes out that catalase, but this certainly wasn't wiped out. You see that? That'll go on until it gets rid of all of the extra oxygen. Now I keep putting more in, it'll just keep doing it. That's extra oxygen bubbling off. But as you move out from the membrane to the edge of the skin, you don't have it out here. It's not there. It's here. It says, no, you can't come in here. You can go out there. We don't care about that. That's just the outside part. That's going to fall off anyway. That's basically your fascia coats you and protects you, and it's always trying to get invaded. And if it can get through and get into here, you're in trouble. So here is where you immediately attack the things that are trying to attack you, and one of them is oxygen. They call it reactive oxygen species. You want to see this thing foam up again? That baby's got some serious amount of catalase. Look at it. Look at that take off. So anyway, that's what I expected from the big artery. But boy, I tell you, that's sometimes you get what you get, not what you need. I guess you get what you need, but not what you want. Or is what you want, but not what you need. It's one of the above. Take your pick. Only one from each column, though. All right, this is my little bone here that's turned into that mud fossil. That right there is the last little bit of bone that's left. Now, coming down, this is the side of the bone. I'll show you this later. But this is coming straight up the bone. Now, coming into this area is where the marrow is. That's marrow. You see that black stuff? That's marrow. All right, the bone is on the outside. This is this side of the bone. And that's the marrow inside. And that's all the way around. The outside of the bone is bone. And you can see it's bone, actually. It's turned to stone for the most part, but you can see there's still bony parts. Now, we're going to get to the part where there's a lot of bone. Well, I say a lot. There's enough we can tell it's bone right there. You see, you come around, there's still bone there. We come around to this part of the bone, and then there's this stalky bone. All right, now I'm going to put a taste of water on there. That always makes things look more, they rehydrate, and then they give off more colors and details. You see it? The rest is black because it's marrow. That's bone. So what we're looking at right there is this right here in the microscope. And here's the lights go on, and here it is. This was that bone. Now that's a bone that is attached to another bone. It has the ligament. So this would probably be in a wrist or something like that. You still got the cartilage, all the tunica. That stuff is tunica, the wrapping of it. And you can see inside where all the, well, I might as well show it to you. All right, here we're going to go. We're going to look at that bone forum in there. Or actually, that's the, where the ligament attaches. There's it right there. Now, I got to turn the lights off so we can see this. Now, I can see the vein in the artery here. Quite easy to see. The red and the black. All right, the The artery is there, the vein is there. Now, I'm going to put a little bit of hydrogen peroxide on there. Just because it's here, I might as well give it a shot and see what happens. I would expect to see a pretty good reaction. And we can see what happens with the red versus the black. Alright? That's a very good reaction. Now, you see the black? It's almost nothing. The red side is going to be bubbling like crazy in the membrane. The black doesn't do it. That's the key. You got to be working with the red blood. You see how active that thing is? That's all a catalase reaction to the hydrogen peroxide. It's very, very reactive. You see? But you don't see a big bubbling coming out of the black side. It's just like dead. It doesn't have any catalase in it, it looks like. I expect something to be going on there. It's not. It looks to me like this probably died laying with this side down. Because this is where most of the reaction's taking place. It appears. Well, actually, no, it's all over. It's everywhere except for the black spot. Almost nothing at all going on there. You learn something every day.